Although there are many exciting applications of working with DNA, such as screening patients for disease, identifying evidence found at a crime scene, looking for relationships between diverse organisms, etc., very often the limitation is the small amount of DNA which can be obtained from a sample. If DNA can be amplified, there can be enough to test, say to run through gel electrophoresis. It is possible to take a double-stranded molecule of DNA, to heat it into single-stranded DNA, and then to add the enzymes such as DNA polymerase, which will synthesize more DNA from this process. Unfortunately, the heat which is required to separate the DNA would then destroy these enzymes. Thus, it would not be feasible to replicate large amounts of DNA in this fashion because of the cost and the labor involved. In the 1980s, work which would later win the Nobel Prize resulted in polymerase chain reactions, or PCR, involving thermal cycling, in which DNA would undergo essentially a three-step process where it would be heated to the point where double-stranded DNA molecules separate to single-stranded molecules of DNA, that primers bind to the DNA sequence of choice, and DNA polymerase then attaches to the primers to then insert the complementary DNA nucleotides to make double-stranded DNA. The breakthrough was the realization that there are organisms such as bacteria which can live in very hot environments, and thus their DNA polymerase enzymes would not be denatured at the temperatures needed to turn the double-stranded DNA into single-stranded DNA. And thus DNA polymerase from an organism which inhabits a hot environment, such as the bacterium Thermus aquaticus, which produces the Taq polymerase used originally in PCR. This can then make the process of amplifying DNA sequences very cheap and easy, because once the DNA is heated to create single-stranded DNA, then primers are attached only to the sequences whose amplification is desired. Now, this allows PCR to be very specific, but also DNA polymerase cannot attach to single-stranded DNA without a primer being present. So this is a requirement for DNA polymerase action. The DNA polymerase from these hyperthermophilic bacteria can then operate inserting the nucleotides from their triphosphate precursors, which are complementary to the DNA strand being replicated. The cycles of separating the DNA strands, attaching primers, and allowing DNA polymerase to replicate the strands can be repeated as often as needed in a programmable thermal cycler, which then could produce thousands or even millions of copies of the desired DNA sequence. Thus, PCR is a technique which is invaluable for modern biotechnology, which amplifies small DNA samples, producing enough DNA where tests can then be run for further application.